Hello, it's my pleasure to talk with you today about work that I did with my former graduate student, Yasmine Akky, and our colleague, Dr. Ryan Earley. Phenotypic correlations among behavior, morphology, and life history traits can inform us about the function of those traits, about adaptive integration among traits, and also about trade-offs, cases where it's impossible to maximize multiple important traits simultaneously. We investigated correlations among these three areas in females of the live-bearing piscillid fish, Girardinus metallicus. This species is endemic to Cuba, where it's widely distributed, and where it co-occurs with a variety of native and introduced predators. We tested the idea that behavior, morphology, and life history traits will exhibit correlations within and across behavioral contexts. With respect to morphology, our primary focus was on a dynamic melanin signal. These are black spots that can lighten and darken in real time, and this is achieved by the movement of melanosomes, organelles that contain melanin. Heathcote et al. Described a dynamic melanin signal in another piscillid recently in guppies. There, darkening of the iris is an honest signal of aggressive motivation. With respect to G. metallicus, the dynamic melanin signal is a dorsal fin black spot. So here is an interaction between a male and a female. The male has static black coloration along his ventral surface, but I'm talking about the dorsal fin spot, which can darken and lighten in real time, and the fin is typically expanded when the spot is darkened. You can see that both the male and the female exhibit this dorsal fin black spot. This spot is also darkened during aggressive interactions. So here are two females fighting with each other. They are biting, chasing, and tail slapping each other. And hopefully you can make out in this video that their dorsal fins are also darkening during this encounter. So we investigated correlations among traits, including this spot, using these general methods. We size match females in pairs, such that we had 32 pairs total. And for each female within the pair, we notched the caudal fin under sedation for individual identification. Either the dorsal or the ventral surface of the caudal fin was notched. Immediately after the females recovered from sedation, we used a non-invasive waterborne method to collect cortisol samples from each female, and we compared the notched females with sham-treated females who were handled and sedated but not notched. And we found no difference in their cortisol levels. The fins regrew within a few weeks after the experiment, and the females seemed to be fine. We tested females 3 to 5 days after they gave birth, such that they were sexually receptive. We also randomized the order in which females experienced the three contexts. So here are those contexts: mating, in which there were two females together with a sexually mature male; foraging, in which the same two females were with a defensible food source, an algae wafer; and a risk context, which involved a female who could take refuge in this compartment or explore a novel environment, an arena that was graduated such that we could quantify the degree of exploration and also the extent to which the female approached a confined predator. This predator was a native Cuban cichlid, Nandopsis tetracanthus, and it was behind a porous barrier. In the first two contexts, mating and foraging, we also scored whether the dorsal fin black spot was dark or light before and after each of 3 trials within each context, such that the female could achieve a score anywhere from 0 to 6 within each of those 2 contexts. Within each context, we scored a number of behaviors. I realize that we don't have time to talk about all of these behaviors right now, but I just wanted you to have a sense of the types of behaviors we scored in each case. Within each context, we subjected the behaviors to a factor analysis to reduce the number of variables and also to uncover latent behavioral factors. We use the scores in subsequent analyses. So here are some of our results. We found a positive relationship between dorsal fin black spot score, so how much it darkened, and aggression from one female to another in the mating context. This spot also darkened in the foraging context, but there was no significant relationship to aggression. So this spot appears to indicate aggression in female-female aggressive encounters. The dorsal fin black spot lightened the more the female followed the male and chased the other female away. So in the mating context, when the female exhibited behaviors that are thought to be mating solicitations, the spot got lighter. So this dorsal fin spot may also indicate immediate receptivity to the male's courtship attempts. With respect to behavioral correlations, the only correlation that was significant is that we found a negative relationship between boldness behaviors in the risk context and aggression in the mating context. Interestingly, there was no such relationship with aggression in the foraging context, suggesting that females are adjusting their aggressive behavior across these two contexts. So to talk about some of the results then that we found. First of all, females were highly aggressive. This is the first time this has been quantified in this species. And female aggression was exhibited using a couple of different behaviors. One that I haven't talked about yet is fin flaring. This is common in fishes. It involves all of the fins simultaneously being expanded for a brief period and then folded again. A fin flare by one female was typically met by a fin fold by the other female. And fin flaring is associated with aggression in a number of fish species. Fin flaring primarily occurred in the foraging context. Darkening of the dorsal fin black spot was the other major indicator of aggression, and this primarily happened in the mating context. This dorsal fin black spot may be an honest signal of both aggression and receptivity to a male's mating attempts. So it's important to note that this black spot is functional in both sexes. It is not a male trait that is merely a byproduct in females, but rather appears to have an adaptive function in both sexes. The dorsal fin black spot also darkens when males are fighting with each other. We found that negative correlation between boldness behaviors and aggression in a mating context. This is in contrast to findings in males of G. metallicus, of other piscileids, and of other taxa. In those cases, with males, typically there's a positive association between boldness and aggression. In females, it's possible that selection actively favors a negative relationship, such that females are either bold and submissive or cautious and aggressive, so that they exhibit an adaptive negative relationship. It's also possible that there's a trade-off. Both of these behaviors are likely to be costly types of behavior, and so there may be a trade-off such that the same females cannot sustain high levels of both aggression and risk-taking behavior. Piscileid fishes have been a very fruitful system in which to examine correlations among traits both within and across taxa. An excellent example of an across-taxon comparison is this study by Resnick et al. From this year, from which I borrowed this image. And traditionally, with the exception of studies of mate choice, behavioral studies of piscileids have largely focused on males. I hope that I've shown you today that studying females can be important and that it can uncover patterns that are interesting regardless of whether they are consistent with what we find in males or stand in contrast to what we find in males. I want to thank these people without whom the study could not have been done, and also want to thank Cal Poly for funding this work. Thank you so much for listening to this talk.